My dear students, today I will start a new chapter, Molecular Basis of Inheritance. Here you will learn some of the basic concepts of molecular biology, that is the central dogma. Before going into central dogma, I would like to tell you about the types of nucleic acid. Now there are two types of nucleic acid. They are the DNA and the RNA. DNA stands for deoxyribonucleic acid and RNA stands for ribonucleic acid. Now both these nucleic acids occur in living system and can function as a genetic material. DNA is a genetic material in all organisms except some viruses. Now this DNA or deoxyribonucleic acid is helically twisted double chain mixed polymer of deoxyribonucleotides, which is not only the largest macromolecule but also represents the genetic material of organisms and molecular basis of heredity. Now, where do this DNA occur? The DNA is an acidic substance which was first isolated by Friedrich Mischer in the year 1869 from the nucleus of pus cell, and he called it a nuclein. In eukaryotic cells, DNA occurs inside the nucleus, and two types of semi-autonomous cell organelles like mitochondria and plastid. So that means DNA is present in all eukaryotic nucleus. Along with it, DNA is also present in mitochondrion and plastid. Now DNA present inside the nucleus is called the nuclear DNA. But the one which is present inside the semi-autonomous organelles like mitochondria and plastid are called organelle DNA. Now nuclear DNA is associated with histone protein to form chromatin fiber, while the organelle DNA is without histone protein. It is termed as naked DNA. Naked DNA also occur in prokaryotes. That is, the DNA is without association with the histone protein. So in prokaryote, DNA containing organelles are absent. Nucleus is absent, so nuclear DNA is absent. Now nuclear DNA is embedded in the cytoplasm and hence it is called chromosomal DNA, as the true nucleus is absent in a prokaryote. Now this DNA may be folded with the help of RNA and polyamines. Many prokaryotes also possess extranuclear or extra chromosomal small circular DNA segment, and these DNA segments are known as plasmids. Now, the chemical components of DNA has been discovered prior to 1950. Two lines of investigations helped in the derivation of DNA structure, that are the Chargaff'S rule and X-ray crystallography. Now what is Chargaff'S rule? Through his work on estimations of various chemical components of DNA, E. Chargaff put forward certain generalizations, and these generalizations are called Chargaff'S rule. Now what are they? They are as follows. Purines and pyrimidines occur in equal amount. Purines found in DNA are adenine and guanine, while pyrimidines of DNA are thymine and cytosine. That is, adenine plus guanine is always equal to thymine plus cytosine. Thirdly, the sugar deoxyribose and phosphate residue occur in equal number. Fourth, the purine adenine is equimolar with pyrimidine thymine. Fifthly, purine guanine is equimolar with pyrimidine cytosine. So the basic ratio is A plus T by C plus G, that is adenine plus thymine divided by cytosine plus guanine, is specific for a species. So it does not change with age or tissue. The ratio can be used to identify the species. Students, you can see here there is an example. How to solve this type of questions? Now you can apply Chargaff'S rule to sort out this type of questions. The question says that in a sample of DNA, if the amount of adenine is 23%, what will the amount of guanine? So you see the given amount of adenine is 23. So obviously the thiamine will be 23%. So 23 plus 23 is 46. If we count the total number to be 100, so the remaining amount is 100 minus 46, that is 54. So the left amount is guanine plus cytosine, that is 54. So you again divide it by 2. 54 divided by 2, that is 27. So the amount of guanine is 27%. So in this way you can solve this type of questions. When one of the any purine or pyrimidine value is given, you can find out the other. X-ray crystallography. Now, X-ray diffraction study of DNA was proposed by W.T. Astbury. It states that DNA is a polynucleotide where successive nucleotides occur at the interval of 3.4 angstrom. Franklin observed DNA to be helical. Wilkins and Franklin obtained very fine X-ray diffraction pictures of DNA which were immediately made available to Watson and Crick. In the next video, I will discuss about the Watson and Crick model of DNA. So my dear student, today's topic was up to this. I have discussed here about the very introductory part of the molecular basis of inheritance, where you have learnt about the Chargaff'S rule and X-ray crystallography.